Guys, what's going on? Today we're going to be talking about glycolysis. Get ready. Glycolysis is so important. We'll find out why. Today we only have Pete. Wallace is in Venezuela. I'm not sure what's going on. We'll find out soon. All right, so where does glycolysis take place? That's the first thing we need to know. It takes place in the cytosol. So again, the cytosol is the fluid that contains our organelles. It's part of the cytoplasm. Okay, and so essentially, glycolysis, the main part is that we take glucose, C6H12O6, and convert it to pyruvate. Now, there are other things that are produced in this metabolic pathway. We have 4 molecules of ATP, but since we are investing 2 in the beginning, which we will go deeper in further, the net ATP we gain out of this process is 2. We get 2 molecules of NADH, energy-rich molecules, useful for in further steps of respiration, and 2 molecules of H2O. All right. So pyruvate is, if you don't know, is an anion of pyruvic acid, which basically is CH3COCOO minus. You don't need to know that. Essentially, what we do need to know is that pyruvate is used in the citric acid cycle. It acts as the link, and it also used to produce lactic acid or ethanol. And the reason we use it for lactic acid and ethanol is in the absence of oxygen, cells undergo fermentation, right? And in doing so, we don't care about the ethanol or the lactic acid. We free up the NAD H, making it an NAD plus molecules that are ready to be used as oxidizing agents because they will be reduced during glycolysis. So with fermentation, we produce 2 ATPs, and although that's not a large amount, it's better than the 0 ATPs we would get otherwise. Again, fermentation occurs with the absence of oxygen. So why do we care about glycolysis? If you haven't realized already, glycolysis is the first step of respiration, and so it's pretty important. All right. So glycolysis, like I've said, is a 2-step beast. We have the preparatory phase, the energy investing, and the payoff phase, where we harvest the energy. All right. So we start off with our glucose, and we have a phosphate group transferred to the glucose to make a glucose 6-phosphate. The glucose 6-phosphate is then rearranged into an isomer called fructose 6-phosphate, and then a second ATP transfers a phosphate group to create fructose 1,6-biphosphate. So after this step, we've already invested 2 ATP. One was going in to make it glucose 6-phosphate, and then another was added to make it fructose 1,6-biphosphate. And then the fructose ring opens, and the 6-carbon fructose 1,6-biphosphate breaks into a 3-carbon sugar phosphate called dihydroxyacetone phosphate, and it's isomer G3P. But as of this part, we want both 2 molecules of G3P. So we have an enzyme that converts our DAP and rearranges it into G3P. So now we have 2 molecules of G3P, and we've finished our energy investing or preparatory phase. Moving on to the energy harvesting or the payoff phase, we have 2 molecules of G3P. They gain their phosphate groups and are oxidized, forming 2 molecules of NADH. So here NAD is being reduced, acting as the oxidizing agent for G3P. As a result, 2 molecules of 1,3-biphosphoglycerate are formed, or BPG. The 2 molecules of BPG transfer phosphate groups to ADP, forming 2 ATPs and 2 molecules of 3PG. The phosphate groups of the 2 3PGs then move up, and we get 2PG, 2 molecules of 2PG. So then the 2 molecules of 2PG lose the water, so that's where we see where H2O leaves, right? And becoming 2 high-energy phosphoenol pyruvates. From here, also known as PEP, the 2 PEPs transfer their phosphates to ADP, forming 2 ATPs and 2 molecules of pyruvate. So from this step, we have created 4 ATPs. 2 when we added phosphate groups to ADP from the biphosphoglycerate, and also 2 when we took the PEP phosphate groups and added it to ADP. And what this is called is substrate level phosphorylation. This will be contrasted with oxidative phosphorylation, but basically what we need to know here is that in substrate level phosphorylation, we are bringing the energy that comes along with the phosphate group when we add it to ADP to form ATP. So then we end up with pyruvate, right? So that's our product, and we've gotten 4 ATPs, but since we invested 2, our net gain is 2, and we've done a lot of other things. But another important point to note here is that we see magnesium, right? And basically, in a lot of these steps, magnesium acts as a cofactor or a coenzyme to help the reaction to help speed up the reaction. And we also have our as our legend tells us, we have our orange steps where the reaction is not reversible. So exergonic, energy releasing. Now, it's important to know that we don't need to start with glucose. We can enter in as a fructose 6 phosphate. So any of these intermediate steps can be the start of our glycolysis. All right, guys. So that's it for glycolysis. In another podcast, we'll be taking a closer look at what sort of enzymes play a role in glycolysis, like our hexokinase and whatnot. But for now, that's all we gotta know. I hope you guys enjoyed this podcast. So I know that was pretty fast. So I mean, watch the video over for retention. As we've all know, repetition is the best form to remember something. So yeah, that's all, guys. The more you know, the better you are. All right, guys. Pete signing out. Peace.